Nervous system, part one. I wanted to share with you a couple of items regarding the nervous system. When we look at our WIDS objectives, we see that the first five examine sensory function, examine motor neural pathways, and describe the role of the autonomic nervous system. From there, we're going to move into the action potential as well as neurophysiology, which will discuss other potentials, impulse conduction, and synaptic transmission. I'm going to make these videos relatively brief. I will be including the links and access to this slideshow as well as the video. Sensory function is first. So when we look at sensory function, we're primarily thinking of nerves. Nerves provide feedback from the peripheral nervous system to the central nervous system. We've looked at a lot of this information in general A&P. So in the Padlet, you're going to want to go back and review some of the videos as well as the link to the website in the first Padlet. So most of this initially is going to be review. So I'll make it brief. Afferent pathway is another term we think of when we're considering the sensory aspects of the nervous system. And we also think of the internal receptors or the interoceptors where you might feel, those are the deeper ones where we'd feel pain, the visceral pain, hunger, nausea, the urge to urinate, those types of things. And then proprioceptor, you'll recall, has to do with position in space. Exteroceptors have to do more with the superficial aspects when we get into touch, temperature, pain, certainly. And then we also have the special senses, so which we spent time on in general. So anyhow, you can go through all of these. There's a sensory function video that you can click on and take a little peek at. That moves us into the second objective, which has to do with motor neural pathways. The motor system, we think of going from the central nervous system out to the peripheral nervous system. So again, with the sensory going from peripheral to central, we now have motor going from central to periphery. And again, that goes back to what we'll see in a few minutes, the stimulus response mechanism. So keep that in mind as we go. That motor pathway we consider to be part of the efferent pathway. Again, afferent is sensory going in, efferent is motor going out. The motor neural pathways are going to begin in the cerebral cortex and travel down through what we call the pyramidal tract and the extrapyramidal tract till we finally end up in the spinal tracts. You'll recall we look at those a little bit, and I have another little illustration to show you those spinal tracts. And again, you can go back into your patent text and into some of those PowerPoints and that information to review, as well as in the notes section of these PowerPoints. When you view them, you'll be able to see some of my own notes that go into more detail. So anyhow, there's a 2-minute cortico-spinal tract video. So again, cortico having to do with the cerebral cortex, spinal meaning we're going down the spine. So just a quick review. Remember, if we see spinal second in a word, that means we're on a motor tract going from somewhere in the brain down into the spinal cord. When we see spino first, spino thalamic tract, for instance. Spino coming first means we're dealing with a sensory pathway going from the spinal cord up to the brain. So spinothalamic would mean spine up to the brain. That's a sensory pathway. Whereas something with the brain, cortico, for instance, and then spinal tells us we're starting in the cortex of the brain going down the spinal cord. That's a motor pathway. Stimulus in, response out. Afferent in, efferent out. Sensory in, motor out. So we have this continuation of sensory and or stimulus and response going out. So anyhow, there's a short video on the tracks, and again, here they are. We can see the motor pathways in red, primarily going to be lateral and anterior, which we saw, I believe I had that, yep, lateral and anterior. So this is going to be the anterior portion of the spinal cord, the lateral portion. And again, these are going to be the pyramidal and extrapyramidal. Okay. And again, we see the word or the combining form at the end, spinal, 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 spinal. That tells us that we are definitely dealing with a motor pathway. We come over here, we see spino at the front. That tells us we're dealing with sensory, meaning we're going from the spine up to the brain. So anyway, a little bit on tracts. So again, probably a review for several of you. Role of the autonomic nervous system is the third objective. The autonomic nervous system, you'll recall, is going to be part of that peripheral nervous system. So the nerve pathway that involves the sympathetic and parasympathetic divisions. So we're going to have motor and sensory sympathetic, motor and sensory parasympathetic divisions. And again, sensory comes in to the central nervous system, motor goes away. Okay. We think of the autonomic nervous system as being divided into sympathetic, parasympathetic, also known as fight or flight, and rest and digest. We relate adrenaline and epinephrine with fight or flight and the sympathetic aspects. We relate acetylcholine with rest and digest and the parasympathetic division. And again, the role is really to help maintain homeostasis, to have a communication pathway for the sensory to work with the control center or the integration center we call the brain, then spinal cord, and then getting that response continually sent out. So again, there's a small or a short crash course video, and I also have other notes in the slides that you can view. We do see that parasympathetic or homeostatic pathway coming primarily from the brain stem, the pons, the midbrain, and the medulla, or midbrain, pons, medulla, or I mean medulla, pons, midbrain. Okay. Pupil contractions, salivary glands, and we get down a little closer to the upper cervical region and into the medulla. We see some pneumotaxic as well as cardiac centers. And we see down all the way in the lumbosacral region the regulation of some of the more visceral, abdominal and pelvic visceral organs. So again, this parasympathetic is the main maintenance of homeostasis. This is what keeps us in that 98.6 degree Fahrenheit, whereas the sympathetic is that fight or flight, the stress response. So we see most of those centers are going to be those descending pathways heading out and traveling to different organs to increase or decrease whatever it might be. So again, we're usually increasing things on sympathetic and decreasing on parasympathetic. So we can read through those as well. The next little objective goes into action potentials. You have maybe heard that term at some point. I know all of you have heard the term nerve impulse. So when we hear of a nerve impulse, we're thinking of action potentials as well. How do we get this sensory traveling to the central nervous system? How do we get the motor traveling away from the central nervous system? How do we get communication to occur from neuron to neuron? And it's through a series of potentials, action potentials being one of them, probably the most well known. So we'll focus right now on that. When a neuron is at rest, when there's no action or no movement, we would say that that everything is polarized regarding these ions or charged particles. We primarily think of sodium outside of the neuron and potassium inside of the neuron. Okay, so when we're at rest, sodium outside, potassium inside, we have this balance of charge. We say that we're polarized. As soon as a stimulus comes in contact with a neuron, primarily with the dendrites of a neuron, we're going to start to see some movement and triggering of sodium going in and potassium starting to come out. So we see when that stimulus comes in, sodium via these stimulus-gated channels is going to start rushing in or diffusing rapidly into the cell. And again, we see this term diffusion, which simply means we have a lot of something going to where there's less of something. So we know we have a lot of sodium out. It's going to diffuse naturally into a cell as soon as those channels open up. That's now what we call depolarization. So we've gone from being polarized at rest and then depolarized, meaning sodium is rushing in. We now have an imbalance of charge. Okay. Generally, we'll see this later in part 2, we're going to see threshold potentials. We're going to see resting membrane potentials. So at rest, we can monitor the movement and the charge of these ions. And at rest, there's no movement. We say it's polarized. We can measure the amount of charge that's on that membrane. And we typically will see, again later, this is getting a little bit ahead, but just as a kind of a teaser trailer, negative 70 millivolts is going to be our resting membrane potential. And again, that number is measuring the amount of negativity that's at the cell membrane. And typically we would think again as that negative 70 means it's highly, there's a lot of negative at that membrane. Sodium is a positive ion, so when the positivity rushes in, it's gonna decrease the amount of negativity. So when I add a bunch of positives to a negative, then I'm going to see the charge increase. So we go from a negative 70 at rest up to, like on a number line, over or up to negative 59. That simply means positives have rushed in and increased the amount of positivity, therefore decreasing the amount of negativity. So we go from negative 70. So think of it this way: we're $70 in the hole in our checking account, and we put $11 into the checking account. We're now at negative 59. So we've increased some positivity, but we're still in the hole. We're still at a negative. There's still a negative amount of charge that will remain always for the most part until at this point, until, like I say, next video, we'll see a change, a rapid change into the positive. So your bank account gets into the positive here in a little bit. So anyhow, thresholds, what triggers an action potential is when we've started to put, so we're at negative 70 at rest, we start putting some money in the bank, it gets closer to zero, it's still negative 59. Once we hit negative 59, however, an action potential gets triggered. And that's going to cause what we see as voltage-gated channels. So the voltage is going to change. That's when we measure, we see these millivolts. Okay. So that's getting a little bit ahead. So if none of this is making sense now, just wait until later. It'll really not make sense. No, I'm teasing. It'll make more sense in the next video. So so just keep that in mind. An action potential just means there's a potential for action to happen, and the only way to get action to happen is to increase the amount of positive ions inside of the neuron. And that happens when sodium rushes in. Okay, we call this, we've got a couple of areas when we deal with an action potential. As sodium rushes in and channels start opening, we're going to see the axon start to get involved. So again, I have a couple of illustrations and images that we'll look at shortly, but again, just the basics of an action potential. Just remember, it's a nerve impulse, and it's going to involve sodium rushing into the neuron. Okay, we'll look at it in more detail next video. There are a couple of items you can watch and click on to understand a little bit more. But again, here's the plasma membrane of the neuron. This is a neuron. So when we look at the plasma membrane at the input zone, or the dendritic zone, this is where we're going to see sodium channels begin to, when a stimulus comes in, a stimulus arrives at the input zone, that's going to trigger some of the sodium channels to open. If we get a strong enough stimulus, those sodium channels continue to open, and they sum together, or they summate. And as soon as we get enough money in the bank right here, so we go from -70 in the bank, as soon as we get to -59 in the bank, so we've added $11 basically, at that point, we're going to see an action potential triggered. So they also call the summation zone the trigger zone. And so that's going to perpetuate or cause the continuation of an action potential, and sodium potassium pumps to be working, and depolarization. And so we'll get into voltage changes in the next video. But this is a neuron. You guys all remember the neuron. We've got the dendrites, the cell body with the nucleus, and then the tail, or the axon. And again, most neurons have multiple dendrites and one axon. Okay, so the input zone. So all of these dendrites are going to be connected to the ends of other axons. And then this axon end is going to be connected to other dendrites from neurons. So the traveling of an impulse begins at the input zone, or the dendrites, goes through the cell body into that axon hillock, or summation, or trigger zone. Once we go from -70 in the bank to -59 in the bank, now we really start to see a lot of money get put in the bank. So we'll see us go up into the positive for a little while here, to really get the fast movement of that stimulus to continue along that axonal tail, heading out the output zone to a synapse, and then another neuron. So anyway, we'll look at that in more detail later. So here's just a quick kind of a preview, the depolarization, repolarization idea. So at rest, when we can measure the movement of these ions, when we're at rest, we can see that sodium is all on the outside, potassium is on the inside. So we have what we call polarized being at rest. As soon as a stimulus comes in, sodium channels are going to open, and we see a rush of sodium going in, and that triggers the initial depolarization. And again, if we put enough sodium into the bank, meaning there's a strong enough stimulus coming in, that's going to trigger voltage-gated channels to open, and then even more money rushing into the bank. That's where we get that -70 in the bank to -59. We're getting a little more. If we get to -59, then we're going to see an action potential generated. Okay, once we hit a peak, we do have a limit of how much money can go in the bank, which we'll talk about again a little bit later. And then we're going to start to see the money go down a little bit. Okay, until we, unfortunately, well, and they call that repolarization. So we're going to see repolarization. And that just means we're going back to being polarized. We're going, unfortunately, back to zero. But again, it's not unfortunate. If it really was a bank, that would be quite the roller coaster. But this is going on perpetually. A nerve and a neuron has to rest in between impulses, simply to basically reorganize all the sodium and potassium ions. So anyway, more sodium rushes in, membrane moves towards a positive direction. Again, if we go from -70 and we start to get more positive in the bank, up to -59. Okay, and after the action potential peaks, and again, we'll see that action potential peaking at anywhere between 30 and 55 plus. So we get out of the hole quite a bit. So we'll look at that in more detail in the next video. Okay, so we do have potentials, impulse conduction, synaptic transmission. So again, we've got these leak channels for sodium, potassium. There's a little bit of leakage constantly going on. If we do get some excitation or a stimulus, we're going to see sodium channels open. When we want to inhibit or stop the impulse, we see potassium channels open. And again, all of this is going on at the input zone or at the dendrites. And then near the summation and conduction zones down by the axon hillock, we do see the voltage-gated channels opening up. So again, we start off at -70 millivolts at rest. We're polarized, and then every positives and negatives are opposite. And then as soon as we get a stimulus, we get a little money to put in the bank, we see once we get -70, -69, 68, 67, once we get to -59. And if we don't, say we get to -60, that means the stimulus wasn't strong enough and everything just stops and we don't get an action potential. The nerve impulse ends at that wherever that space was. So anyway, once we do get -59 in the bank, now we see an action potential develop, and then we get all the way up to a +30 in the bank. So a pretty good from -70 up to a +30. So we've got about 100 voltage change going on there. Once we hit that peak, we start to see the process of repolarization. So now money's going back out of the bank as we head back to being at -70 or being polarized. Okay, and then we'll look later on at there's a brief moment of hyperpolarization where we dip even a little deeper past our -70 mark in the bank. But that's a very short period of time that involves a little bit of juggling of all this sodium being pushed back out of the cell or being pumped back out of the cell. We get a little too negative toward the end, or we pump a little too much out. So those leak channels will allow some of that sodium to leak back in, and that will enable us to be polarized. Yeah. And again, we've got the resting membrane potential is -70 millivolts. We do see those sodium leak channels at the local potential, more in that getting into the summation zone of the cell body of the neuron. We see the stimulus-gated channels as well as at the dendrites of the input zone. Those get open as we travel closer to that summation or that hillock of the axon. That's where the threshold potential in the bank may hit that -59. So again, when we look, let me go back. There it is. So again, when we look at this is that input zone, resting membrane potential. We start to see of -70 as we increase the number of positive ions in, we start to see, we get to what they call a threshold, which is again negative 59, and then threshold potential. And then that negative 59 will trigger the action potential, as well as voltage gated channels, which are larger channels that are going to allow a lot of sodium to then rush in. That's why we go from negative 70 to negative 59. We kind of creep with these stimulus gated channels, but as soon as we hit negative 59, we get the floodgates open and our bank account shoots up really quickly. So that's an action potential. And again, sodium outside, potassium inside. The negativity inside is primarily caused by these anionic negative proteins. So again, when we measure that negative 70, we're taking that voltage right at the membrane. So what we end up seeing is a lot more negativity at that spot. So as positives rush in, we see less negativity. So this goes up a little bit to negative 59. And again, resting membrane potential, nothing happening. Okay. Typically around negative 70 millivolts. We think of those sodium and potassium leak channels. We get a little bit of leakage. We also have some sodium potassium pumps. And whenever we see the word pump, we generally are thinking of ATP or energy being necessary. So the sodium potassium pumps are going to pump 3 sodiums out of the cell and 2 potassiums back in. So it's that 3-to-2 ratio, and we'll look at that in more detail. Okay, so once I get sodium, excess amounts of sodium in the cell, the sodium potassium pumps are going to get to work to start pumping the sodium out and bring the potassium back in. Okay. And again, this is going to be occurring really all of the time, but especially, once we're going through that repolarization, we're starting to see our bank account go back down. That means we've started pumping sodium back out of the cell. So when we go from negative 70 to negative 59 up to positive 30, that means a lot of sodium is diffusing in. That does not take any ATP. So sodium rushing in during that action potential from negative 59 up to positive 30, that sodium rushing in doesn't take any energy. You can see the word diffusion. Diffusion just means we have high concentration to low concentration movement. Okay. Once we get to plus 30, we peak out with the sodium. Now we start going back down. But in order to get the sodium out, we need to implement some ATP and that sodium potassium pump. Okay. And again, ATP, you're thinking glucose, oxygen, mitochondria. Those are all going to be glucose, oxygen, mitochondria. You're going to get the maximum ATP. That's why sugar consumption or glucose consumption, hydration are extremely important. Breathing is extremely important, as you know. Okay. So ATP. The brain and the nervous system as a whole are extremely reliant on glucose. Okay. So the local potential, again, once we start shifting from the resting membrane potential of negative 70, we see a stimulus or some excitation. The stimulus gated channels open up. Again, we're still at the input zone. Excuse me. If we get to a threshold of negative 59, then we're going to see the voltage gated channels open and an action potential occur. We don't need to discuss those. We can look at what an action potential is going to look like. We see that negative 70 resting membrane potential. A stimulus comes in, it excites the stimulus gated channels for sodium to open. So we see a little bit of sodium coming in. If the stimulus is strong enough, and we get to negative 59, At that point, an action potential is generated, and voltage-gated channels open up and allow for more sodium to rush in until we get to about a plus 30. And then at that point, voltage-gated sodium channels close and the potassium channels open. Potassium pumps are going to start working to pump sodium out. And then finally, we get to back to -70, get all the voltage-gated potassium channels to close up after that hyperpolarization. And again, there's a little bit too much sodium potassium movement until we hit a threshold there. And then that's going to trigger about -80, and that's going to trigger the potassium channels to close. Then we're back to -70. We'll look at wave summation and refractory periods in one of the physio X's we do for muscles. So a lot of what we're seeing today and in this chapter, we're going to see throughout the semester. So anyway, refractory, absolute refractory, spatial summation, trepi. We'll look at some of that with muscles, and that'll get into these different types of action potentials that we may see. The thing is about an action potential, it's all or nothing. Let me go back to this slide here. Once it starts, once we get to here, it has to go through this process. Okay. If it doesn't make it to here, we can drop back down. So if I add a bunch of stimuli, I have to get one, I can summate them together. So we're going to see this, or sum them together. We'll see this in one of our physio X's, like I said. Okay. Nerve impulses. Again, myelin is important. Fastest nerve impulses typically move at 130 meters per second. Slowest at about 0.5. And again, we need that myelination. You remember that from general A&P. Myelin is like an insulator that allows for nerve impulses to move fast. Okay, so we're going to see electrical synapses between neurons, and we'll see chemical synapses between neurons as well, but also with neurons and muscles where we involve neurotransmitters. So that pretty much takes us to the end of part one. Part two gets into neurotransmitters a little bit. We'll continue to talk about synapses really throughout the semester. But again, we see the electrical synapses with the gap junctions. And again, just a change in charges. These are going to be from neuron to neuron oftentimes. The pre-synapse and post-synapse. And then over here, we see more of a typical chemical synapse where we have chemicals stored and we release them into the post-synapse. So the synapse is usually an axon terminal, or the end of an axon, meeting up with a dendrite or meeting up with a muscle cell. Okay, so a synapse is any nerve and nerve meeting up. So a dendrite, an axon and a dendrite coming together, or we could have a nerve and a muscle. We have nerves and glands meeting together, but whenever a nerve ends and something else begins, we call that a synapse. Okay, so you have a synaptic knob, and then the cleft is the connection of the synapse. Again, the action potential reaches that synaptic knob, but that's going to trigger the dendrite of the next neuron to at the input zone to then start potentially, depending on the strength of stimulus, to get the stimulus-gated channels open, and then the voltage-gated channels open until we get down another axon to another dendrite, and so on. Okay. So anyway, I'm going to wrap it up. There's some of this is we'll see throughout the semester, but again, you can see a chemical synapse. This is very normal with a muscle cell, so we'll see that in a couple of weeks. We've got the pre-synapse and the post-synapse, or the axon terminal and the potentially a muscle cell. Okay. So that's it. If you ever have questions, again, email me. I will post this, and I'll be posting the PowerPoint as well, separate from the video. Part 2, I'll record and upload that shortly.